Welcome to Lecture 10, where we will go over plant structure and function. We are now going to take a look at how a plant grows. Plants have what is termed indeterminate growth. This means that they can grow continuously throughout the lifetime of the plant. This is due to the presence of undifferentiated cells in regions called the meristems. These cells can continuously divide and differentiate. This is different than determinate growth, which is present in most animals and certain plant organs, such as the leaves, thorns, and flowers. Determinate growth stops growing once they hit a certain age or a certain size. These regions of indeterminate growth are located in the apical and lateral meristems. The apical meristems contribute to the vertical growth of the plant. Thus, they are located at the roots as well as the apical or axillary shoot tips. Lateral meristems are responsible for the secondary growth or thickness of the plant. These consist of cylinders of dividing cells that run along the length of the roots and the stems. In addition, vascular cambium and court cambium are also important in the growth of the plant. Vascular cambium adds vascular tissues called secondary xylem and phloem, while the court cambium replaces the epidermis with the periderm. Once cells are displaced from the meristem, they can divide multiple times before they start to differentiate or specialize. During what is termed primary growth, these cells can differentiate into three primary meristems. These primary meristems act as primary tissues that will eventually form mature tissues. These areas are called the protoderm, ground meristem, and procambrium. The protoderm will eventually form the dermal tissue. The ground meristem will eventually form the ground tissues, and the procambium eventually forms the vascular tissues. Please take a look to the image to the right to note the difference between these different primary growth tissues as well as their location. Note the differentiation as they mature into the dermal, ground, and vascular tissue. This slide depicts the role of the apical meristems in the primary growth of the plant. Note that the root apical meristems are located at the tips of the root. Also note that there is an apical meristem located at the top of the shoot or top of the plant. When taking a look and examining the growth of the root system, note the presence of the protoderm, ground meristem, and procambium. The protoderm is located in blue that eventually differentiates and specializes into the root hairs as well as the epidermis. The ground meristem forms the cortex of the root, and the procambrium eventually forms the vascular cylinder. Note that these tissues originally result from the root apical meristem, which is the group of undifferentiated cells. These cells will then proliferate and start to differentiate into the three different meristems. Once this zone increases, this is called the zone of elongation. Eventually these cells will begin to differentiate, and this is noted in the zone of differentiation. These will then take on the cells of their final form, cells of the root hair, epidermis, cortex, or vascular cylinder. This slide depicts the primary plant growth process in the shoot. This is present in the above portion of the plant and results in the vertical growth upwards. Note that the apical meristem is located at the top. This is typically surrounded by leaf primordia, which eventually will form leaves. Cells within the apical meristem will divide. These cells are considered to be undifferentiated. When they divide, some of them will remain in the apical meristem so that it is replenished. However, some of those daughter cells will not. The daughter cells that leave the apical meristem region will eventually differentiate into different tissue types. As a daughter cell becomes partly differentiated, it will move along the length of the plant. This contributes to the growing of the plant. As these cells move further away from the apical meristem, they will go further along in the differentiation process. This slide depicts the process of secondary growth in plants. Secondary growth is made possible by two lateral meristems. These meristems will extend along the entire length of the shoot or root. Secondary growth occurs when primary growth has ceased. These lateral meristems are called the vascular cambium as well as the court cambium. The vascular cambium will eventually differentiate into vascular tissue, while the court cambium will eventually differentiate into the epidermis and eventually the periderm. These lateral meristems consist of cylinders of dividing cells. When these cells divide, they will then undergo a growth period. As these cells grow, they will increase the circumference, thus contributing to the secondary growth or thickness of the plant. The apical bud refers to the apical meristem that is located at the very top of the plant. This is responsible for the vertical growth. The apical bud will have bud scales that help to protect the important meristem or the group of undifferentiated cells. During the spring, these scales are shed and allows for new growth to occur. New growth will occur at both the apical bud as well as the axillary buds that are located on the tip of the branches. This will help to produce nodes as well as internodes. Scars will remain at the previous growth sites. Take a look at the image to the left to see the difference in the 3-year growth of a tree bud. Note the presence of the leaf scars, which remain at the previous growth sites, as well as the bud scars. Note how the stem is growing in the vertical direction and the branches are growing in the axillary or lateral direction. Even though plants have meristems that have indeterminate or continuous growth, they can still die. Flowering plants are usually classified based on their life cycles. They can be annuals, meaning they complete their life cycle within 1 year, biennials, meaning they require 2 growing seasons to complete their cycle, or perennials that can live multiple years. We're now going to take a look at the growth process in roots. The entire biomass of the primary root derives from the root apical meristem. The primary root typically has a root cap. This serves to protect the apical meristem and the cells underneath. This also allows for the root to penetrate into the soil. Growth in roots typically has 3 major zones. The zone of cell division, which includes the stem cells and all of their immediate descendants that result from mitosis. The zone of elongation, which is the region of growth. This is when cells are continuously being added to the younger end. As they travel through the zone of elongation, they begin to differentiate into the protoderm, ground meristem, and the procambium. Finally, the zone of differentiation, or the zone of maturation. In this region, cells completely differentiate into their specific cell types. As the zone of maturation increases, the root elongates and new cells are added to the growing root tip. When examining the primary tissues, the protoderm will develop into the epidermis and into the root hairs. This is comprised mostly of epidermal cells. The ground meristem will eventually form the cortex or bulk of the root. This is comprised mostly of parenchymal cells along with the endodermis that will form the boundary with the vascular cylinder. The procambium eventually forms the vascular cylinder. This is comprised of xylem and phloem tissues surrounded by a pericycle. This image shows the difference in the arrangement of a typical eudicot and monocot root. Note in a eudicot root the presence of xylem and phloem in the centralmost region of the root. This will then be surrounded by the pericycle and vascular cylinder. Beyond that will be the endodermis, cortex, and eventually the epidermis. In a typical monocot root, the parenchymal cells make up the central region. This will then have xylem and phloem interspersed throughout the parenchyma. This will then be surrounded by the pericycle and vascular cylinder. This will then be surrounded by the endodermis, cortex, and eventually the epidermis. Just like the upper part of the plant or above ground part of the plant can branch, the roots can as well. Lateral roots can form as outgrowths from the pericycle. These outgrowths can eventually push through the outer tissue or epidermis and emerge from the original root. This can help create extensive root networks for the plant. When looking at the process of primary growth in shoots, the entire mass of the shoot derives from the shoot apical meristem. This includes the leaves and stems. The apical meristem is a dome-shaped region of dividing cells that is located at the shoot tip. As stated previously, this is usually protected by leaves of the apical bud. The shoot apical meristem will produce protoderm, ground meristem, and procambium. Lengthening and division of these cells in the internode region will cause the shoot to elongate and grow upwards. This image depicts the differential arrangement in eudicot and monocot shoots. In a typical eudicot shoot, the vascular bundles of xylem and phloem form in a ring-like organization. The ground tissue is then divided into the pith or inner portion and cortex or outer portion. Note the presence of the vascular bundles or phloem and xylem in the eudicot shoot. Also note the presence of fiber cells that offer support to the vascular tissue. This is then surrounded by the epidermis. In a typical monocot shoot, the vascular bundles of xylem and phloem are usually scattered throughout. The ground tissue is not partitioned into the pith and cortex as we see in eudicots. When considering the overall growth of a plant, there is what is termed apical dominance. This means that the apical bud usually has top priority for growth within the plant. This means that the buds that are closest to the apical bud are usually inhibited from growing. This is usually controlled by chemical communication that occurs within the plant. However, if an area is grazed, this control can be disrupted. This allows for compensatory growth or also for pronounced lateral growth. When the apical meristem is grazed or cut off, the lateral shoots will then take over to contribute to the lateral growth of the plant. We're now going to examine the growth of the leaf. Leaves develop from the leaf primordia and are a result from the primary growth process. This means there is little to no secondary growth or adding or contributing to the girth of the leaf. The dermal tissue will form the upper and lower epidermis, the guard cells, as well as the stomata. The epidermis has a waxy cuticle to once again prevent desiccation. Guard cells will surround the stomata to help to control the opening and closing, to also control the dissipation of water, heat, and the exchange of gases. Ground tissue within the leaf forms the mesophyll. This is made of parenchymal cells and bundle sheath cells. In eudicots, there are two layers of mesophyll, the palisade and spongy mesophyll. The palisade mesophyll will consist of one or more layers of elongated cells that are very rich in chloroplasts. The palisade layer resides immediately underneath the epidermis and is adapted for harvesting the most amount of light as possible. The spongy mesophyll contains irregularly shaped cells. These lie just above the lower epidermis. This layer is specialized for the movement of gases. As a result, there are many air spaces that are present within the spongy mesophyll. The vascular tissue extends throughout the leaf as well. Veins will continuously divide within the stems and leaves and branch throughout the mesophyll to ensure that all plant cells have access to nutrients and water. Xylem is responsible for bringing water and minerals to the photosynthetic cells, while phloem brings the sugar and organic products produced by photosynthesis to the other parts of the plant. Bundle sheath cells surround and protect the vessels and also help to regulate the flow of material throughout the leaf. This diagram depicts the general organization of the leaf tissue. Note the presence of the stoma or openings that open and close due to the presence of the guard cells. These are located in the upper and lower epidermis. Under that is the palisade mesophyll, which contains the elongated cells that contain the chloroplasts that carry out the bulk of photosynthesis. Below that is the spongy mesophyll that has many air pockets that helps to regulate the exchange of gases. This is then enclosed by the lower epidermis. Also note the presence of a waxy cuticle on the upper and lower epidermis. Within the mesophyll, palisade and spongy, there will be bundle sheath cells that contain the xylem and the phloem. These will help to distribute water and minerals, and as well they will carry the organic compounds made by photosynthesis to the rest of the plant. The secondary growth in plants usually occurs in the lateral meristems, and remember it contributes to the overall plant thickness. Secondary growth occurs primarily in gymnosperms and eudicots and is very rare in monocots. In addition, secondary growth occurs in the stems and roots and rarely occurs in the leaves. The vascular cambium adds the secondary xylem and phloem, while the cork cambium produces the waxy cells that protect the plant from water loss and also helps to protect invasion from insects, bacteria, as well as fungi. Primary growth adds length to the stems and roots and helps form the leaves, while the secondary growth increases the diameter of the plant. This final slide depicts the primary and secondary growth in a typical eudicot plant. Note that the primary growth occurs in the apical region. Also note that the secondary growth does not occur until the primary growth is almost done. Also note the differentiation of the different tissues, specifically the xylem, the phloem, the cambium, as well as the pith, cortex, and epidermis. Note that as secondary growth occurs, you get the deposition of the bark and the different layers of periderm. This contributes to additional support for the plant that supports the vertical growth.